Humans like to put everything into neat little categories. And so we're always trying to classify new organisms that we find. We started out, I should say, by looking at the structure. What did the organism look like? And then we started looking at, okay, how does it work? How does it metabolize? Now that we know about DNA and RNA, we can actually use genetic fingerprints in their DNA to help us classify the different organisms. And so the largest classification is the domain. There is a video that I'm going to show you the link for. I'm not going to play it on here because it doesn't sound really good. I just tried it and now I'm remaking this entire thing because the video looked horrible. So I've added the link over here. You can either copy and paste this link, or you can just go into your Google bar and write McGraw-Hill 3-domain animation and it will pop up. The 3 domains are separated based on DNA and RNA fingerprints. So they're looking at the characteristics in the DNA and RNA to see how closely related different organisms are. And so what they figured out is that eukaryotic organisms are slightly more closely related to Archaea. Bacteria and Archaea are even closer related to each other than they are to eukaryotes. Okay. Once we have put each organism into a domain and classified them into the proper categories, we then want to give them a name. The type of name we give is a tune naming system called scientific nomenclature. It was produced by Carolus Linnaeus, and this is how we name all organisms. We use the genus of the organism, and then we use the specific epithet, which gives us the species name. So most of you have heard of E. coli. E. coli is the shorthand version of Escherichia and coli. So the species name is always written with the genus name capitalized and the specific epithet lowercase. So Escherichia coli is written, and then we're going to either underline the name or we're going to italicize the name. So in eukaryotes, we have a lot of categories under the domain. We have the kingdom, which takes in all the organisms that fall under a specific group. So it's the most inclusive. It includes almost everything. And then we go down to the phylum, class, order, family, the genus, and then the species name. Under bacteria, we don't have the kingdom. We have the domain, and then we go down to the phylum, class, order, family, genus, and species. There's a lot of mechanisms that we use to classify microorganisms. We initially use the structure of the organism. We run tests to identify the shape of the organism. But once we have that, we can use biochemical testing and genetic testing to help us to identify the organism down to its actual species. And one very helpful mechanism to identify organisms, microorganisms, specifically bacteria, is a dichotomous key. So a dichotomous key is a key used that has two answers to any given question, and it helps us to follow test results. So this dichotomous key starts with some bacterial species. We run a test called a gram stain, and in the gram stain, we can either get positive or negative results. If we get negative results, which this looks like it was, then we can do another test. This would be a chemical test to determine if they utilize lactose. We can get positive or negative results again. And in this case, we got positive results. So if we have positive results, we can then run another test to see if they produce indole from tryptophan. So if we have positive results, then we can use a citrate test to see if they can use citrate as their sole source of carbon. If it's positive, then they are this species. If it's negative, then they are this species. Over here, if you had negative results from the indole production, you can then run a methyl red test to see if they are a mixed acid fermenter or if they are not. So if you're positive, you're Citrobacter frundi. If you're negative, you're Enterobacter aerogenes. So this is how we do a dichotomous key, and in class I will teach you how to make a dichotomous key, and we will actually be making one together from that wonderful sheet that I gave you that has all the characteristics of your microorganisms. In microbiology, we don't always, in fact, we can't see the organisms that we want to study without the use of instruments like microscopes, because most microbes are smaller than the eye can see. So we use microscopy to look at the tiny structures. In our class, we will be using light microscopes to look at bacteria, and possibly we will also look at some helminthic organisms that are more macroscopic, but we can see structures inside of them. So the light microscope is used to identify organisms in our class, and in general, it's a really effective mechanism to look at most microorganisms. It's not effective at seeing viruses. So viruses, you have to actually get a more advanced microscope, like an electron microscope. So with a light microscope, you're going to use light, which will pass through the lenses and the specimen. And what we will see the microorganism under the highest power, which is oil immersion, so that we can identify the microorganism, its shape and its size. But we can only do that if we stain the microbe. So microbes are basically colorless unless we stain them. So a stain provides color or contrast to the microbe. So we color the microbe, and then we can see the difference between the microbe and the slide itself. There are different types of stains depending on the type of microbe that we're working with. A simple stain uses a positive or a basic dye. The dye is going to be attracted to the negative proteins in the bacterial cell. And so the dye adheres to the cell wall and it sticks there, staining the cells but not the background. In a negative stain technique, we use an acidic dye that is repelled by the bacterial cell, because in biology and chemistry, opposites attract. Proteins in the cell wall are negatively charged. So if we use a positive dye, that positive charge is going to be attracted to the negative cell wall. But if we use a negative dye, the negative dye is going to be repelled by that negative cell wall. And so with a negative dye, we actually stain the slide, but we don't stain the cells. We have some special stain techniques that allow us to know more than just the size and shape of the bacterial cells. In a gram stain, we're able to identify if bacteria are considered gram-positive or gram-negative. So this is a chemical difference in the bacterial cell wall. Gram-positive bacteria have a thick layer of peptidoglycan in their cell wall. Gram-negative bacteria do not have that thick layer of peptidoglycan. They have a very thin layer of peptidoglycan. So in a gram stain, we use basic dyes, so positively charged dyes. We use a primary dye called crystal violet. Then we add iodine. Iodine acts as a mordant, basically glues the crystal violet in. We then use acetone to get rid of any excess color on the slide. And if you're gram negative, because you have a thin layer of peptidoglycan, that acetone will rinse the cell completely clear. So there will be no stain left in it. Then we do safranin as a counterstain. And only cells that have no stain in them will be stained by the safranin. So we end up having gram-positive cells being a purplish color and gram-negative cells being a pinkish red color. We have some very specialized stains that can help us to distinguish specific microbes. So we can use in an acid-fast stain, identifies bacteria under the genus Mycobacterium, because Mycobacterium have an acidic structure called mycolic acid. It's like a waxy structure, and that waxy structure keeps, or I'll say keeps, that's a good word, keeps other stains from being able to set into the cell. So they don't stain well in general. But we use a special technique that helps the carbol-fuchsin to stick into the cell. And then we rinse it with an acid alcohol and then counterstain with methylene blue. So only acidic cells, I won't say only acidic, only Mycobacterium cells will keep that carbol-fuchsin inside the cell, and they turn this beautiful pink color while all the other cells will be blue. And the reason this is such an important test, even though it only looks for one genus, is because Mycobacterium produces two very serious diseases, tuberculosis and leprosy. And if we can identify the bacteria rapidly, then we can start to treat these disorders. Oh, you know what I didn't talk about? Okay. One other stain technique that we will be using in lab is an endospore stain, which helps us to identify cells that can persist in the environment. So remember in chapter 1, Tyndall and Cohn identified these cells that were heat resistant. Those cells were called endospores. There's an endospore stain that you will learn that identifies not just one species or one genus, but it identifies any organism that produces endospores. So we can find those persister cells in a group of cells. And you'll learn this as well in the coming weeks. We can use other microscopes, but they cost a lot more money. So there's dark field microscopy, there's phase contrast microscopy, there's electron microscopy. In each case, these are a lot more expensive, and we don't have these microscopes. So you're not going to actually get to use these. But I will mention that in phase contrast microscopy, we're going to be able to see living organisms that are unstained. We just don't have the ability or the means to do that. In dark field microscopy, we're looking at the background is going to be very dark and the cells will be light. It's almost like doing a negative stain. And then I didn't mention I talked about electron microscopy, but I didn't really go into that. There's two types of microscopy that are electron microscopes, transmission, and then scanning electron microscopes. These can see very small viral particles, so super tiny. All right, this is the end of this chapter. So this is a pretty short chapter. We're going to get into chapter 4 next, which is a longer chapter that deals with the structure of prokaryotic organisms versus the structure of eukaryotic organisms. So you have a wonderful day, and I will see you soon.